Good night. My name is Sebastian Ceron L. I'm on third semester of Universidad Pontificia Bolivariana, and I'm going to expose about the development of a rapid antiviral screening assay based on eGFP reporter virus of Mayaro virus. Okay, so as an introduction, we need to talk about the virus Mayaro, who is an arthropod-borne virus that is mainly transmitted by Aemegogus mosquitoes. The virus can be transmitted from non-human primates to human primates when they are close in proximity to the forest. The Aedes aegypti mosquito, which is responsible for the transmission of Zika and Zika virus, has been shown to transmit Mayaro virus in experimental tests. GFP, along with proteins derived from other organisms, offers an inhibitor of fluorescence proteins, among other characteristics to make it without a doubt special. The fluorescence protein stands out for not needing intermediaries for expression in any living organism. Okay, the general objective of the investigation is the eGFP expression level could reflect viral replication well. Using the known inhibitors of alpha virus, we showed that Mayaro virus eGFP could be a powerful tool for antiviral screening. Different support means can be used to avoid mechanical disturbance and the effects of convection currents on separation. As support media, paper, cellulose acetate, strong gels, agarose gels. These methods are used for transfection, a chemical, physical, or biological method. The transfected nucleic acids may stay transit or stable in the cell, depend on the method of transfection. Nucleic acids transfer into eukaryote cells, as we said. Fluorescence said that it is a histochemical laboratory staining technique used for demonstration the presence of antibodies bound to antigens in tissues or circulating body fluids. These techniques are essential to supplement clinical finding and histopathology in the diagnosis of immunoglobulin disorders. They permit early diagnosis, treatment, and subsequent monitoring of disease activity. The reverse transcription is the opposite process that we know that is copying RNA to DNA. The single-stranded RNA is transcribed into single-stranded DNA, but the RNA-dependent DNA polymerase enzyme, and it's called reverse transcriptase. The reverse transcription is commonly used in research in a technique called reverse transcription polymerase change reaction (RT-PCR). With the help of our RNA can be transcribed into DNA. We are going to talk about the characterization of the recombinant virus, which with the immunofluorescence assay of viral protein expression in BHK21 cells transferred with the Mayaro virus RNA. The immunofluorescence assay was performed at the indicated time points. Transfection using the antibody against the E2 protein. Here we can see how at 14 hours there are not too much antibodies that are shown. At 36 hours and at 48 hours we can see there are too much antibodies shown and react with cells of the construction and characterization of Mayaro virus eGFP reporter virus. First, in the V image, we are going to analyze the eGFP expression in the BHK21 cells transfected with Mayaro virus eGFP RNA. The expression of eGFP was detected under a fluorescent microscope at the indicated time point after transfection. So here we can see how at 15, 24, 39, and 48 hours the cells react to the eGFP virus but with the transfection, and we can see, and in the image C we can see how the immunofluorescence assay analysis of viral protein expression in the same BHK21 cell transfected with the Mayaro virus and the Mayaro virus eGFP RNAs respectively. So here we can see the Mayaro virus but with eGFP and without. And here we can see how at the time in the image A we are going to see the eGFP expression of the different passage of Mayaro virus eGFP in BHK21 cell. The Mayaro virus eGFP was serially passed in BHK21 cells for 5 rounds. So here we can see how does it increase in each image. In the image B, we are going to see the detection of the eGFP gene during virus passage in BHK21 cell. Total RNAs from the infected cells were extracted and subjected to RT-PCR detection that include the complete eGFP gene. So here we can see how they are at the same time and they are equal. We can see it, and just 2 of them are different. In the image C, we can see the eGFP expression of the different passage of Mayaro virus eGFP. So here we can see the C6, about 36P1, P2, and P3. And here we can see that the P1 is the highest antibodies in cells have reacted with them. In the image D, we can see the detection of the eGFP gene during virus passage. So here we can see they are at different level, they are not at the same. The optimization of the Mayaro virus eGFP infection for antiviral assay, we are going to see also that the eGFP gene expression was detected under a fluorescent microscope at the indicated time points after infection. So here we can see that MOI at 0.01 in 12 hours, they are like anything. And we are going to see how to plus the concentration of MOI and the hours it increased 24 and 36. Discussion were to talk about Hotel and Moore said that in recent years our viruses including dengue, Zika and chikungunya had re-emerged and caused several worker outbreaks. I'm in agreement with him because we have seen that those 3 sicknesses, dengue, Zika and chikungunya, had re-emerged, and we need to help people and create vaccines and to prevent that stuff. August had said that the constructions of the infections clones of different viral strains will be helpful for evolutionary and ecological analysis of Mayaro virus. And he is right, and I'm agreeing with him because this construction of the infections clones help to detect the antibodies and how to create the vaccines and prevent those sickness. Holtz and Murray has said that as a mosquito-borne virus that threatens human health, Mayaro virus is a typical neglected, that there are few studies of this important pathogen and in the stinging literatures. I'm also agreed with him, and this reconnects because there are not too much studies about the Mayaro virus, and this investigation helps to know more this virus and to prevent sickness. So as a conclusion, the use of the GFP protein has been used as a tool to help and detect vaccines that are very useful in the medical file. As we talk of the discussion, this GFP protein help to detect the antibodies and to create vaccines, and also it helps people. And the transfection helps in the introduction of external genetic material into eukaryotic cells by means of plasmids, which helps in research to study the genetics of organisms and their response to certain change. The eGFP, that it is a fluorescence protein, that it is expressed in any living organism, and its use in biochemistry, microbiology, genetic engineering, and physiology. It helps and detect vaccines and disease. Thanks for your attention.